Hi, sorry to bother you, but I was wondering, what do you do here at Stroud?
Oh, hi. Well, I'm a geneticist here at Stroud, at the Stroud Water Research Center. And I study fish and mussels and things that are related to the heredity of DNA and questions about the evolution of fish species in streams and rivers.
So how do you find the DNA of these critters?
Oh, that's an excellent question. There's a couple ways we can do it. If we can't capture the individuals, we can actually get their DNA from a water sample. So it turns out that animals are sloughing off cells all the time. And so cells will enter the water column and they'll float downstream. And if we capture that cell in a water sample, we can look for the DNA that way.
Oh, okay. So how do you get it then if it's in the stream?
Oh, well, that's an interesting question. So in order to get it, we have to get into the water with a bottle. And we have to make sure that we have a very clean bottle, so we don't want any contamination. We don't want anybody else's DNA in this bottle because any contamination could confuse our results. We don't know where that DNA came from. So we start with a very clean bottle, and then what we do is we go in the stream and we dump the bottle in the water and we lift it out, and we take that water sample back to the lab, and then we filter it. And so we capture the little cells, the little particles that are floating in that water. It's actually that filter is where we get our DNA.
Oh, okay. So how do you know then that you're capturing the DNA of the mussels, just the mussels?
Okay, so yeah, one of our important questions here is we're looking for freshwater mussels. And so we don't know if they're present in White Clay Creek. We know they used to be here. They used to be very abundant. Stories say that you could actually walk across the stream on the backs of mussels. Sounds painful, but but that's what they say. Mussels, we know, are very important pieces of the ecosystem. They serve an important role for filtering water. And so, what was the question again?
Sure. So the question was how do you know you're finding mussel DNA and not, let's say, a fish?
That's an excellent question. And so in order to be sure that what we're finding is just a mussel DNA, is we look at the DNA sequence. And so we actually know, so the DNA sequence, that's the A's, the A, C, G, T. And so the order of those A, C's, G's, and T's is specific to the freshwater mussel. And so we look for that sequence that only the freshwater mussel has and nobody else has. And so if we find it, then we can be pretty sure that we've got a freshwater mussel in our stream.
Awesome. So have you had any problems with your experiments or any issues that come up with eDNA, since you said it's such a small molecule?
Yeah, so our protocol is so sensitive we could potentially detect one copy of DNA. And so like I said before, any contamination could be misconstrued misinterpreted as a mussel that was present. And so yeah, we have to be very careful to not contaminate our samples. And it could happen anywhere along the chain. So unfortunately it does happen, but we also use negative controls throughout our experiments to make sure that we aren't getting any contamination.
Awesome. Well, thank you so much for your time. I'll let you get back to reading.
Thank you. And good luck.
Thanks.